Memories associated with traumatic events tend to be vivid and extremely difficult to forget. And experiencing a traumatic event sometimes might lead people to develop something that's known as post-traumatic stress disorder, or PTSD. In fact, not all people who experience the same traumatic event develop PTSD. So people who experienced 9/11, for example, only about 20% of them went on to develop PTSD. 80%, however, still had strong memories of the event, but did not have the anxiety associated with it. Why some people are more resilient or susceptible to anxiety disorders has been a long-standing question that has remained largely unresolved. And this is where basic scientists like me come to play. In my work, I use mice models to study this disorder. So I try to model aspects of post-traumatic stress disorder by exposing mice to traumatic events and subsequently testing them for the memory of the trauma. Now, while I was testing these mice for the memory of the traumatic event, I realized that not all mice display fear behaviors, just like not all humans who experienced 9/11 develop PTSD. But unlike these humans, mice are nearly genetically identical, the mice that we study. They have essentially the same life experience and they just underwent the same event. Yet one group differed dramatically from the other. This offered me an opportunity to ask and search for a fundamental biological mechanism that might underlie the susceptibility to trauma. Cells in our brain, called neurons, are connected in a very precise manner to each other to form networks, and memories are stored in these networks in the form of pattern of activity. We found a way to measure and quantify such changes in these circuits, essentially being able to visualize a memory trace. I started studying these memory traces in a particular group of cells called cholinergic neurons. I realized that mice that do not display fear behaviors, even after experiencing trauma, have defects in reactivating this memory trace. I followed this finding with a few more experiments that suggested to me that indeed these cells and their circuits are in a position to tune the extent of the traumatic memories. So, why do we care? After all, this was all just in mice. Well, it turns out that circuits that process threats are largely evolutionarily conserved. And so this does offer us an opportunity to test a new system now as a target for PTSD. But beyond that, I would like to say that even today, people view psychiatric illnesses such as anxiety disorders as a moral failure or a character shortcoming. And I hope that studies like mine can help inform people to add legitimacy to people who are suffering with these illnesses and also give hope to people who are suffering with these illnesses to go and seek treatment. Thank you.